Hello everybody, myself Dr. Anjana Anand from Common College for Girls Sector 14 Gurugram, and I would be taking topic cytokinin, its history of discovery, biosynthesis and mechanism of action. Now the name cytokinin is derived from the word cytokinesis, which means cell division. That is, cytokinins has something to do with the division of cells. They are a class of phytohormones which promote division of cells, and cytokinin, besides cell division, has a role in nutrient management, uptake of nutrients from the soil, or reallocation of nutrients within a plant, and particularly during senescence. And many aspects of plant life are related to cytokinins. And regarding the structure, cytokinins are N6 derivatives of the nitrogen purine base adenine, which are characterized by their ability to stimulate cell division in tissue culture. Kinetin is the first cytokinin discovered, and it is a synthetic one and does not occur naturally, but was originally synthesized from the herring sperm DNA. And zeatin is the most widespread naturally occurring cytokinin in higher plants. All cytokinins have basic adenine structure. In this picture, we can see that N at the top is the N6 side chain that is an important part of making a cytokinin. Now this slide shows different structures of cytokinin. They are zeatin, isopentenyl adenine, kinetin, and 6-benzyl aminopurines. As I said earlier, naturally occurring cytokinins are adenines, which possess either an isopenoid or aromatic side chain at the N6 position. Now, the isopenoid side chain of isopentenyl adenine can be hydroxylated in either the cis or trans terminal position and form zeatin. It is named according to its first discovery in maize. The double bond of zeatin is reduced in dihydroxyzeatin, but a reduced equivalent of isopentenyl adenine has not been detected in plants till date. The isopentenyl or isopenoid cytokinin are widespread in nature, whereas the aromatic ones are solely synthetic. In this slide, we can see the formation of active forms of cytokinins, isopentenyl adenine and transzeatin. Both of these cytokinins are produced by the removal of ribose groups. Now, for the production of cytokinin, we need ribose adenine and pentenyl group together, and slowly with the removal of ribose sugar, functional kinetin is formed. Now we'll discuss about the discovery of cytokinins. There are various paths, that is the path investigating cytokinin, the discovery process, and the role of Miller in purifying the factor from herring sperm DNA, and also the use of yeast extracts. Now, this Cook's lab, they used coconut milk and for finding the factor which was responsible for inducing the shoot formation. Now, growing plants in culture is not easy. Contamination or the plant growth conditions and very harsh sterilization protocols. These are all the major factors which are to be faced in the tissue culture conditions. Now, in the Cooks group, they source the coconuts from the local grocery store, and they found that the coconut milk only occasionally induced shoot formation. And the Skook sought advice from an expert in the use of coconut milk in cell culture, and he also sought help in the purification of the milk to make it more consistent for his studies in shoot formation. At this time, Skook did not care uh, about identifying the factor in coconut milk that induced shooting, and he just wanted a way of making shoots. But professor at the Cornell University, his name was F.C. Stewart, and he responded that only his lab would be using the coconut milk. And this rebuff it sparked Skook to compete with the Stewart to find out the factor in coconut milk that induced cell proliferation. And Scoop contacted his colleague, Frank Strong, he was from the Department of Biochemistry, to collaborate with him in this effort. Now, the members of both the labs they developed purification methods and important bioassays to test extracts at each step of the protocol. Now, the Carlos Miller, he also entered. He was a postdoc in Skoog's lab. Now, Miller, he also set to work in improving the culture medium for growing the plant tissues. He also worked to identify the growth factor that was causing the cell proliferation and shoot formation. And one thing he tried adding to the growth medium was a bottle of yeast extract. He thought it might enhance the auxin activity in the medium. Uh, Medium yeast extract, as yeast extract had compounds similar to the auxin. And he happened to choose an old bottle of yeast extract, and it turned out to have some of the growth promoting activity. Now, Miller had thought it could be a source of auxin activity which induces rooting. A new batches of yeast extract was also used, but it did not cause the shoot promoting activity. Now, he worked with this one special bottle, and he found that this bottle of yeast extract has something to do with the shoot inducing activity. And based on some previous work from the Skoog's lab, and adenine had weak shoot inducing activity also. And Miller tested a bottle of herring sperm DNA. He found that similar to the coconut extract and his special bottle of yeast extract. That both herring sperm DNA, its bottle of yeast extract, and the coconut milk all had the capability to induce shoot. Now, Skoog was excited by his findings and he ordered a keg of herring sperm DNA for the lab. And however, like his new bottle yeast extract, the new herring sperm DNA also showed no shoot inducing activity. That is only in one of the previous bottles there was a factor in which caused the shoot activity. And Miller was able to partially purify the factor from this original bottle of herring sperm DNA, and he was able to, or we can say he was able to achieve about a concentration of 1 milligram per liter of water. And he found that this factor was quite potent, like the ethylene and auxin. Miller had discovered that preparation of herring sperm DNA left at room temperature for months developed shooting activity. Now it was also found with the yeast extract. Now he found that this factor also promoted the formation of shoots, and they even predicted that it was an N6 substitution structure that was responsible for shoot inducing activity. Now, this is the story, or we can say that this story tells us that yeast extracts or you can say the coconut milk or the herring sperm DNAs, these all had the factor that induced the formation of shoots, and the factor was found to be N6 derivatives of adenines. Here we can see the picture of Carlos Miller, Frank Strong, Fox Cook, Francis Okamura, and Malcolm de Zalza. And it was the beginning for the first isolation of a cytokinin kinetin in 1955. This team had worked on tissue culture a lot in 1950s. Functions of cytokinin: it stimulates cell division and morphogenesis in tissue culture. It also stimulates the growth of little buds by the release of apical dominants, stimulates leaf expansion resulting from cell enlargement, and it helps in maintenance of undifferentiated callus growth. We know that callus is an unorganized mass of cells, and it is achieved with equal molar concentrations of auxin and cytokinin. Now, high auxin to cytokinin ratio encourages the development of root, whereas if it is inverse, it encourages the development of bud. By the exogenous application of cytokinin, to detached leaves will delay the onset of senescence. It will also maintain the level of proteins, and it will prevent breakdown of chlorophyll. It will also delay senescence in intact leaves. But when there is a natural senescence, there will be a decline in the amount of cytokinin which is exported from root. Here is the layout of functions of cytokinin. Cytokinins are responsible for morphogenesis, for differentiation, for enlargement of cells and cell division, and it leads to delay in senescence, nucleic acid metabolism, and slow down the process of aging and activate protein synthesis. Now this is the slide which shows how the relative concentrations of auxin and cytokinin have major functions in the formation of a complete plantlet. So if we can see here that if the concentration of auxin is very high as compared to that of the cytokinins, it forms root on the cuttings. And if we go on the lower side, there is somewhat inhibition in auxin concentration and the rise in cytokinin concentration, which leads to the formation of root on callus. Here we can see that if we go to the lower side, there will be callogenesis, and by the decreasing concentration of auxin and increasing of cytokinins, and then the formation of adventitious buds, and lastly the shoot proliferation. Now, cytokinins also influence morphogenesis in culture tissues. We discussed it earlier also. The maintenance of undifferentiated callus growth is achieved with roughly equal molar concentrations of cytokinin and auxin. And high molar concentration of cytokinin to auxin will induce the development of bud. But if it is inverse, as we discussed earlier, it will encourage the development of root. It is thus possible, by manipulating the balance of cytokinin and auxin in the medium, to generate a complete plantlet, and it can be formed from the undifferentiated callus tissue in sterile culture. Now these plantlets can be transplanted into soil in the greenhouse or in the field, where they can grow into fully competent mature plants. This capacity to regulate morphogenesis in culture tissue has found a major role in horticulture and agriculture, and it is referred to as the micropropagation. And by this, tissue culture can be used to produce large number of virtually identical clones, which would be viral free in a very short time. In this slide, we can see that cytokinins control root vascular development. Now, the major site of cytokinin biosynthesis in higher plants is the root, and high cytokinins level is found in roots, especially the mitotically active root tip, and in the xylem sap of roots from a variety of sources. Now, in the root vascular cylinder, we can see in this picture that some cells in the procambium differentiate into protoxylem cells. It can be seen with the yellow arrows, others into metaxylem. It can be seen by the yellow arrowheads, and others into phloem, and seen by the red arrowheads. And we know that it is uh, that the cytokinin level in the root is very high, and these are transported via the xylem to the upper part of the plant. Now, indirect support for this conclusion is provided by the observation that excised leaves from many species can be maintained in a moist sand bed only if adventitious roots are permitted to form at the base of the petiole. If these roots do not form or are removed as they form, the leaves will quickly undergo senescence. So the delay in the senescence is partly due to cytokinins, which are synthesized in the root and transported to the leaf through the vascular tissues. In this slide, we can see that cytokinin is required for normal growth of the shoot apical meristem. The above picture is of wild type where shoot apical meristem is shown, whereas the picture below shows that cytokinin oxidase expression reduces the size of shoot apical meristem and the number of dividing cells. Here we can see that cytokinins translocate through the xylem and phloem. That is, the phloem sap contains isopentenyl adenine and transzeatin. And different cytokinins are produced and transported differently throughout the plant, and these different forms may convey different informations. In the xylem sap, it mostly contains transzeatin and transzeatin riboside, which is synthesized at high levels in the roots by this enzyme CYP735A. Here we can see that cytokinins contribute to nutrient uptake and allocation. That is, if the levels of nitrate and phosphate is elevated, there is increase in the rate of cytokinin synthesis, and which ultimately decreases the root growth rate. Whereas if cytokinin level is elevated, it represses nutrient uptake. And here we can also see that elevated cytokinin levels increases the expression of photosynthetic enzymes and delay leaf senescence. In this picture we can see the source, that is the photosynthesis, which provides photosynthates, and it is in the form of sucrose, and there is a source sink relationship between the uh, photosynthase and the products of photosynthesis. By this slide, we can see the cytokinins delay leaf senescence, and we know that senescence is the process of aging or deterioration of leaves, and it is dramatically reduced in kinetin treated leaves or the leaves of plants which produce high levels of cytokinin. And we can see here the control plants and the plants which produce cytokinin. We can easily differentiate between them. A major route for removal of exogenously supplied cytokinins in many tissues is oxidation by the enzyme cytokinin oxidase, and it is an enzyme that catalyzes the irreversible degradation of cytokinin phytohormones that are extremely necessary for growth, development, and differentiation of plants. Now, this cytokinin oxidase, which is partially purified from tobacco tissue, maize seed, and corn gall disease, it cleaves the isopentenyl side chain from either the zeatin or isopentenyl adenine or their ribosyl derivatives. Here we can see the effect of cytokinin oxidase. B is the wild type where there is normal activity of cytokinin, and in A, due to the overexpressing of cytokinin oxidase, there is greater formation of root as compared to that of the shoot. That is, the activity of auxin has increased as compared to that of the cytokinins. Now I will speak something about receptor signaling aspects. As we know that all hormones must have a receptor, and without a receptor hormones will not work. And however, the cytokinin signaling pathway is somewhat different from that of the auxins. And as in binding of auxin degrades the repressor, there is a transcription factor in auxin which will work only when repressor is degraded. Whereas in cytokinin, its pathway is somewhat similar to that of the bacterial receptor. As bacteria has a two-component system, kinase system, whereas plants and higher animals have kinases, extracellular domains that can receive signal and transmit signal downstream by the activation of kinases, and those are serine or tyrosine or threonine kinases, whereas bacteria uses histidine and aspartate, and histidine here is activated. Here we can also see that it consists of histidine kinases and a response regulator. It is a two component system and is a short signaling pathway that moves information from an input to an output. As in bacteria, it usually consists of two proteins, a histidine kinase and a response regulator. Whereas I said that bacteria has a two-component system, that is of histidine and aspartate. Histidine is activated by phosphorylation, and this phosphate is transferred to the receiver domain of another molecule which has aspartic acid. Or we can say there is a relay of phosphorylation, that is a phosphate group, from the input to the output domains. This is a summary of perception and signaling pathway by cytokinins. Cytokinins are perceived by hybrid histidine kinases, as we discussed earlier. Cytokinin binding initiates phosphorylation, that is the transfer of phosphate group, which ultimately activates type B response regulator transcription factors. Cytokinin signaling is negatively regulated by type A and type C response regulators. Cytokinin signaling is also mediated by cytokinin response factors, transcription factor like B type ARRs. Here we can see cytokinin action in whole plant processes. Cytokinins regulate root vascular tissue development, as we saw in the earlier slides, shoot and root apical meristem, and in functions, cytokinin helps in nutrient uptake and its allocation to different parts. It delays senescence and in many other processes. Now, cytokinins are synthesized by the condensation of an isopentenyl group with the amino group of AMP. Here we can see in the this flow chart that cytokinins synthesize, they are catabolized, they can be conjugated and then transported. And we know that cytokinins will also form conjugates with sugars and are metabolized by the oxidation processes. Now, there are two principal routes for regulating the cytokinin activity levels: by removal of cytokinins from the active pool, conjugation with either glucose or amino acids, and oxidation. Now, glucose conjugates are formed primarily at the nitrogen in position 7 or in position 9 on the purine ring, as we saw in the structures of cytokinin in the previous slides. Now, the N-glucosyl conjugates are very stable and do not appear to be hydrolyzed readily to give the active free base, and their formation thus appears to be more a means of inactivation of cytokinins than for storage purposes. Whereas O-glucosides, they appear to be storage forms that are readily hydrolyzed to yield biologically active cytokinins when it is needed by the plants. And then cytokinins are also transported by the receptors, and we have seen that by the activation or inactivation of the transcription factors, they target the genes, and then the final is the biological functions which cytokinins performs. Here we can see that cytokinins, they provide opportunities for agricultural improvements. As in the first picture, we can see that cytokinin signaling is necessary for development of symbiotic nodules, as they help in the nitrogen fixation. Whereas in the another picture, we can see that if cytokinin level is elevated, the plants are more drought tolerant, and as drought induced IPT expression confers the drought tolerance. And in the lower one also we can see that the watering, 1 week rewatering, there is no development of the plant, whereas in the another one, if the elevated cytokinin levels are treated with the more watering process, then also the plants are better than that of the those which have the low levels of cytokinins. Now, in addition to the effects, these effects, exogenously applied cytokinins will influence a number of other developmental phenomena, and they will stimulate cell enlargement in many of them by the mechanisms as discussed earlier. And by at last, we can say that the cytokinins have a greater role in major developmental processes in plants, and they can be a boom for the in agricultural improvements. Thank you.